Hi guys, we are back again, and today we are going to talk about meiosis. Well, meiosis is a type of nuclear division in which we are taking a cell which has 2N. Now 2N would be diploid. What does diploid mean? Diploid means it has two sets of chromosomes. So we are during this meiosis division, you end up the one cell will produce 4 daughter cells with N chromosomes. That means this is haploid. Haploid is same as saying one set of chromosomes. So basically what we are saying is meiosis, during meiosis cell nuclear division, a cell, one's mother cell, which has 2N, divides first to form 2 daughter cells with N, that is haploid number of chromosomes, and then this will divide again by in meiosis 2, the second half of meiosis, to produce 4 daughter cells, each with N number of chromosomes. All right. So basically, meiosis is a reductional division. Now, why is meiosis necessary? Why can't we use mitosis, be the cell division process that runs sexual reproduction? Because meiosis is found on where sexual reproduction occurs. All right. So here, and what would happen if mitosis was used instead of meiosis for sexual reproduction? Well, we have 46 chromosomes, 23 pairs. So imagine you have a sperm which has 46, and then you have an egg which is also 46. Well, when you unite this, you'll end up with a zygote which is 92, right? And of course what happens is 92 will now produce, the next one will be 184. There is no reductional, which will produce 368, and so on and so forth, right? Instead, we have meiosis. Now watch, during meiosis what happens? Meiosis, you end up with haploid sperm and egg, N and N. When they unite to form a zygote with sexual reproduction, you end up with 2N. So meiosis sort of balances out and helps us keep the number of chromosomes constant. All right? So we are going to the stages, and remember, like I said, meiosis has, so the one daughter cell, this is one daughter cell will first divide to form 2 daughter cells. Okay, so we are talking about prophase 1. Now remember also, before prophase starts, remember the cell has been through an interphase. And what is interphase? Interphase, there are 3 divisions, right? There are 3 stages, excuse me, G1, S, and G2. So here what happens, S, remember, is the DNA replication phase. So chromosomes are already duplicated before we start prophase. So the first thing that happens in prophase 1, which by the way is the longest phase, is the first thing is pairing of homologous chromosomes. What are homologous chromosomes? Homologous chromosomes are chromosomes that carry the same gene. So you have this chromosome from your father, let's say chromosome A from your father, then you have chromosome A from your mother. So they are carrying the same genes. So this is pairing will take place, all right? Now synapsis takes place. Synapsis means that here these 2 non-sister chromatids, so this is sister, this is non-sister, this and this will touch each other at points. So watch what I'm doing. Here is one chromosome of one pair, and here is another chromosome, which I'm trying to draw a little different, of another pair. So here, non-sister chromatids, this and this, hopefully you are seeing that this is different from this, is touching. And this is called synapsis. Synapsis means that non-sister chromatids touch at points. Now crossing over takes place. At these junctions, at these points where they have touched each other, now exchange of genetic material takes place. So look, the tip of this red chromosome went here, the tip of this blue chromosome goes in here. This is called crossing over. Now here, after crossing over, they do not separate. So this picture is slightly faulty. These homologous chromosomes are still stuck. Remember, this has got this tip, and here is my other chromosome of the other pair, and this has a straight tip. So they are still stuck, and this is called a tetrad. Tetrad means 4. 1 strand, 2 strand, 3 strand, 4 strand. This is called a tetrad. Tetrad means 4. So crossing over occurs. This allows exchange of material between the father and the mothers, or the homologous pair of chromosomes. Okay? And of course in prophase 1, remember what always happens: nuclear membrane breaks down, nucleolus breaks down, the centrioles move to the 2 opposite poles, spindle fibers have started to appear, and of course chromosomes are very condensed. So all those things are also happening in prophase 1, which I said is the longest phase of meiosis. The next phase is metaphase 1. Well, metaphase 1, the tetrad, remember it is not chromosomes, the tetrad, the homologous pair which have exchanged chromosomes, they come to lie in the equator or the middle of the cell. And the centrioles produce the spindle fibers which are attached right here to the centromere. Okay, so this is the centromere of the tetrads. Anaphase. Now what happens is that the spindle fibers contract, and just like the puppets are pulled away by these strings, just like that, the tetrads are separated. So each end is getting 1 chromosome of the homologous pair. Okay, so a good analogy would be if you take a black shoe and a black shoe, or a red shoe and a red shoe, I've separated them. All right, so here, and then finally of course we have the telophase. Telo comes from the word tail. This is the last phase, and here you are now seeing that the nuclear membrane is reforming, spindle fibers have broken down, there are no more spindle fibers, nucleolus is appearing, and the chromosomes are slowly starting to become uncondensed. All right, so here also you notice that the early phase of cytokinesis is setting in. The cells are getting pinched, the cytoplasm is going to get pinched, and as you can see, the cytokinesis, this cell is going to separate out, and you're going to get 2 cells, but only have one set of chromosomes. They have haploid. These cells are haploid. They have only 1 of the pair of chromosomes. Okay, so the chromosome number is halved. Now we're looking at interkinesis. Interkinesis, so here we have a cell, divides into 2 daughter cells, and remember, before it goes into the second division, so this is meiosis 1 and meiosis 2 producing 4 daughter cells. Before we go into meiosis 2, there should be a period of rest, and this period of rest is not called interphase, it is called interkinesis, because here no DNA replication occurs. Let's go back, let's remind ourselves. Interphase, this interphase, which happens before a cell divides, has 3 parts: G1, S, and G2. The S phase is where DNA replication is going to happen. In this case, the chromosomes are already replicated. You see them like this, right? Only thing is that crossing over has taken place, so they look kind of a little different. They don't really look, I'm just drawing it like this. So because the chromosomes have already duplicated in here, we don't need to go through all 3 phases of interphase. Yes, G1 will occur, G2 will occur, but not S phase. So this is why this is not called interphase, this is called interkinesis. This happens between meiosis 1 and meiosis 2. All right. So now let's see again prophase 2. Prophase 2 is very much like mitosis prophase 1, or the prophase of mitosis. So very much similar, nuclear membrane breaks down, chromosomes are becoming condensed, nucleolus disappears, that's number 3. And notice that centrioles have moved to 2 opposite poles, and spindle fibers are being formed slowly. You can see them as asters, and here these microtubules are forming, and these are going to attach right here to the centromere of the cystochromatids. All right. That is prophase 2. Metaphase 2. Metaphase 2 again is very much like mitosis. The chromosomes, the spindle fibers have attached to the centromere of these duplicated chromosomes. The sister chromatids are joined right here, remember. This is the centromere. So they attach to the centromere, and just like a puppet is moved with the help of strings, the microtubules or the spindle fibers have moved the chromosomes to the equator, to the middle of the cell. Now comes the anaphase. Anaphase, what happens is that the sister chromatids, they get pulled by the chromatid. So the sister chromatids, remember this one has squiggly ends, this gets pulled, and you get one is like this, one is little squiggly, so they get separated. One will move the one sister chromatids. Once it separates, it's called chromosome. So these are sister chromatids, these are chromosomes. The chromosomes move to the opposite poles. And notice also with anaphase, furrowing has started, pinching of the cytoplasm has started. So we start with telophase, and cytokinesis follows. Here what happens is that nuclear membrane reforms, nucleolus reappears, chromosomes are going to get uncondensed, and the spindle fibers have all broken down. So that is telophase, the tail phase, the last phase. And of course telophase and cytokinesis follows. Cytokinesis follows, which is nothing but the pinching of the cell. And because of this pinching, this furrowing, which happens in an animal cell, you end up with 2 daughter cells. So altogether in meiosis, you end up with 1, 2, 3, 4. And 4. And of course, these cells that are produced are either your sex cells, either your sperm, of course, or your egg, because meiosis is needed for sexual reproduction. That's why whatever the products are going to be gametes. So these are all gametes, okay? So this is just a continuation. Cytokinesis has occurred. You can see the furrowing. And as a result of which, like I said, meiosis, you start out with a diploid mother cell. The cell divides once first and gives rise to haploid daughter cells. The haploid daughter cells divide again in meiosis 2 to form 4 daughter cells, which are gametes. And these cells, remember, are going to be different from each other and different from the mother. And this is how you see genetic variation. Hopefully this presentation has been helpful to you guys. If there are any questions or concerns, please stop by my office, and I thank you for your time and attention.